Synaptic transmission. Yes, for this synaptic transmission, need to learn the basic patterns of neurons here. The two neurons I'm drawing one after another. There you can understand this area. The gap between the two neurons, which can be termed as synaptic cleft. The gap between the two neurons is said to be synaptic cleft. In general times, people use it to say that only as a synapse in actually, but exactly the space between the two neurons. Space means the dendrite of one neuron and axon terminals of another neuron. That particular space can be called synaptic cleft. Before the synapse, whatever the neuron is present, that we say presynaptic neuron. Neuron wise, we use the term called presynaptic neuron. Then postsynaptic neuron, presynaptic neuron, postsynaptic neuron. The axon terminal, the membrane of axon terminals just before the synaptic cleft is termed as presynaptic membrane. Presynaptic membrane. Then the membrane of the dendrite after the synaptic cleft can be termed as postsynaptic membrane. The exact definition of the synapse will be this box. This box can be called synapse. A synapse include presynaptic membrane of the axon terminal, synaptic cleft, the gap which is 200 angstrom length gap is and the postsynaptic membrane is collectively called synapse. But in general time, the cleft whatever we are saying, that cleft is itself considered as a synapse. No matter here. Now what we are looking forward, that particular box we are seeing. Now whatever the box we are looking for that particular box, we are going to draw in a little bit bigger, which we are going to show that in microscope. Already this is microscope. Okay, so this particular area, which we are going to show in microscope, let me say once again, presynaptic neuron and postsynaptic neuron. Then always the impulse will pass from the presynaptic neuron to postsynaptic neuron. Moment of impulse is always from the presynaptic neuron to postsynaptic neuron. Yesterday I said one more thing to you, that moment and impulse is always away from the cyton. Now we are moving again that with a new concept, that movement of impulse is always from the presynaptic neuron to postsynaptic neuron. Then what is synapse? A presynaptic membrane of the axon terminal, the postsynaptic membrane of the dendrite, the dendron. Then the gap present between the synaptic cleft is collectively considered as a synapse. They are collectively considered as synapse. Okay, but in general, the synaptic cleft only we are using it as synapse. Okay, now let us draw this particular area in microscope here, presynaptic membrane and postsynaptic membrane. So in presynaptic membrane, who is present? Axon terminals are present here. Axon terminal are telodendrites. We use it to say axon terminal or telodendrite. I think we just need to reconnect back this. Screen need to connect it back this. Okay, so presynaptic membrane, postsynaptic membrane, and the synaptic cleft I drawn little bit clear for you. And the presynaptic membrane, synaptic knob we say, axon terminals or telodendrites are having synaptic knobs. Okay, synaptic knob. This is the postsynaptic membrane. Here it is. Then the particular axon terminals of the neuron, let me draw again that you can understand that the axon terminal, synaptic knob we can say, that we are showing in microscope. then it contains neurotransmitter containing vesicles are present here. Neurotransmitter containing vesicles. Till the time we are seeing that influx of sodium, efflux of potassium for the increase of the charge and decrease of the charge respectively. Now what is required? Whenever the impulse is passing and moving continuously towards the axon terminals, that when it reaches here they open calcium gated ion channels. Here they are going to open calcium gated ion channels. The calcium gated ion channels are getting open, and that brings the calcium to get inside, and the calcium will come and bind with these vesicles. Excuse me. Calcium binds with vesicles. They bring the vesicles to move forward towards the end of presynaptic membrane, and they make the release of a neurotransmitter called acetylcholine. The neurotransmitter in general, acetylcholine we are taking. There may be some more else like sympathin, like norepinephrine, dopamine. There various other neurotransmitters are also present. We are taking example as acetylcholine. This particular acetylcholine, which is released after the breakdown of vesicles, this acetylcholine that influence the postsynaptic membrane. The postsynaptic membrane is having ligand-gated ion channels. The postsynaptic membrane is having ligand-gated ion channels. We are seeing till the time sodium-gated ion channels and potassium voltage-gated ion channel. What is this ligand-gated ion channel? Ligand-gated ion channels are flexible, that based on the requirement and the place of the body, they make influx of sodium ion, or either they make efflux of potassium ion. That means that place, based on the place that will happen, either influx of sodium ion, both won't happen, that influx of sodium ion that raises the charge to increase, as we know. Efflux of potassium ion that decreases the charge below the polarization, isn't it? So when these conditions, both conditions every time won't happen here, but I am showing everything in one place. So ligand-gated ion channels, if they allowed influx of sodium ion, that create excitatory postsynaptic potential, or else or we are putting R here, that is very important. Or if efflux of potassium ion occur, that makes inhibitory postsynaptic potential, that create inhibitory postsynaptic potential. Nothing, no need to remember, but just for general knowledge sake, we are talking about the state textbook. We have two words like EPSP and IPSP in our state textbook, that when it comes to that NCRT point of view, the sequence is enough here, that presynaptic membrane potential, opening of calcium gated ion channel, calcium gated ion channel binding with the vesicles, and the release of acetylcholine into the synaptic cleft that influence the postsynaptic membrane. That's enough for us. That this is the case synapse here. Let us see a video to understand much better. This is the synapse. Okay, so let me continue with the video.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced. A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane.
Okay, then this is about
voltage-gated channels open and
the action potential, the synaptic transmission we can see. Okay, so then we'll continue with the sense organs tomorrow. We continue with the sense organs, eye and ear. That we'll go for explanation and followed by the animated videos to understand much better, because that's going to be a bigger area, much bigger area in examination point of view. Okay, thank you.